Lab 13, Exercise 41, Antimicrobial Sensitivity Tests. You should be on page 353 of the lab manual. We'll now be looking still at microbial control, but now we'll be looking at the use of basically antibiotics, which are a type of antimicrobial. You should read pages 353 through 364. So page 353, what are antimicrobial drugs? They are therapeutic chemical compounds that kill or inhibit the growth of microbes. There are several kinds of antimicrobial drugs. Antibacterial drugs, who are very specific and that their job is to target or kill bacteria cell structures. So in other words, these antibacterial drugs, they target specific things like the cell wall, the ribosomes, nucleic acids of different bacteria. So basically what you're looking at is we're going to look at those for our methods of actions on the next few slides. Antiviral drugs, which are also a type of antimicrobial, they target or kill viruses. Recall that you do not use antibiotics to destroy or kill viruses. In other words, if you were ill with a viral infection, you should not take an antibiotic. So we're going to get more specific. Antibiotics are compounds produced by certain species of bacteria and fungi that inhibit the growth of other microbes. You will explore this in more depth in the history of antibiotic video, but let me just mention a few things. If you think about this, bacteria have been on this earth before humans. And one of the reasons that bacteria are able to evolve and adapt so easily is because they have developed mechanisms to compete with other microbes. Specifically, bacteria and fungi are able to secrete chemicals into their environment that kill other microbes. We have then taken those chemicals into the laboratory setting and produced or manipulated them to produce different drugs. 99% of most chemicals secreted by bacteria we cannot use to make antibiotics because they are too harmful to our systems. So only about 1% of those chemical compounds can be then manipulated to make drugs that are effective for us. So what we call naturally occurring antibiotics are antibiotics that the bacteria produce without us manipulating them. Semi-synthetic antibiotics, we have taken a portion of the natural state and a portion of our own chemistry in the lab and manipulated it to help us. Alexander Fleming, which you will see and hear lots about in the video, discovered the first antibiotic, which was penicillin. Penicillin is the antibiotic. However, penicillin came from the mold Penicillium. You'll notice, based on what we've talked about earlier in the semester, that Penicillium is the mold that is capitalized because that is the genus and italicized. Specifically, it was Penicillium notatum, the genus and species. You can also see in this picture Fleming's experiment. He swabbed a plate with some bacteria. The bacteria happened to be Staphylococcus. He then went away for the weekend, not realizing apparently he had not used ASAP technique, because when he came back, by accident, some Penicillium, the mold, had been allowed to grow on the plate. He then noticed the inhibitory effect, zone of inhibition, that the mold had on the bacteria. In other words, the mold had secreted a chemical that then inhibited the growth of bacteria. This was the beginning of antibiotics. So let's look at this table of antibiotics. These are some drug families. So what that means is categories of antibiotics. We will look at the categories, the examples, and the method of action. Similarly to the antiseptics and disinfectants, you will need to know how to match up or be able to do in a multiple choice question on a quiz the antibiotic with the method of action. That's how does the drug actually work. So we have our first category, penicillin. Penicillin works by targeting the cell wall. So by targeting cell wall synthesis, we're saying the drug prevents the bacteria from making a cell wall. You should recall from the lecture portion of the course that the cell wall's job is to act as a protective divider or barrier for the cell. So if its divider or protective barrier is not able to be built, then the cell will die. Penicillin Glycopeptides and cephalosporins all have the same method of action, which is targeting that cell wall. This is also one of the most common methods of actions that antibiotics use. This is because we as humans do not have cell walls. We would like to make antibiotics or drugs that are not going to interfere with our own cells. And since we don't have a cell wall, these would not target our human eukaryotic cells. So penicillin examples include penicillin G, ampicillin, and amoxicillin. Glycopeptide example includes vancomycin. Vancomycin is a very powerful antibiotic. That's what we call one of the big guns and should only be used when absolutely necessary. Next we have quinones, whose job is going to target the nucleic acid synthesis. In other words, interfering with DNA and RNA from being produced. Examples include ciprofloxacin, rifamycin, or rifampin. That particular antibiotic is also used to treat tuberculosis. Macrolides, phenocols and tetracycline, their method of action is all to target protein synthesis. Again, this would be translation, because you should recall from the genetics unit, translation is the genetic process that produces a protein. So they interfere in different ways. It might be the ribosome, it might be the binding of the polypeptide chain, it just kind of depends, but they all interfere in some form of way in protein synthesis or translation. So macrolides include erythromycin and azithromycin, which you might know as a Z-Pak. Phenocols include chloramphenicol. Tetracycline examples are tetracycline and deoxycycline. And last but not least, we have our non-penicillin derivative, or in other words, non-penicillin drugs, which are called semi-synthetic. They're made half in the lab and half from nature. An example is a sulfa drug. And again, if you're allergic to penicillin, they might give you sulfa drugs. These drugs target the folic acid synthesis, or what we call essential metabolic or metabolite synthesis of cells. And examples include trimethoprim sulfamethoxazole, all of which, or most of which, of these antibiotics will be tested on the next few slides. Exercise 41, antimicrobial sensitivity, page 354. So broad spectrum antibiotics. So what are they? Broad spectrum antibiotics are antibiotics that treat gram-positive and gram-negative bacteria. In other words, these antibiotics kill both gram-positive and gram-negative bacteria. So let's think about why that's a good thing or not a good thing. An example of this is amoxicillin. So ideally, if you are ill for, let's say, a urinary tract infection, your doctor or nurse would take a sample of your urine and do what's called a culture. After they get the results back of the specific type of bacteria that's causing your urinary tract infection, they should then prescribe the most specific antibiotic to treat your infection. So for example, if E. coli is the bacteria causing your UTI, they should give you an antibiotic that treats E. coli, which is gram-negative. Broad-spectrum antibiotics such as amoxicillin, as we've seen, are going to kill both types of bacteria. Therefore, they would kill the good bacteria that's supposed to be in your body and the bacteria or the pathogen causing your illness. This can also lead to antibiotic resistance because you're killing the good bacteria in your body, and in females, this can also lead to yeast infections because you're killing both good bacteria and the pathogen. So it is better to take a narrow spectrum antibiotic. This is an antibiotic that treats gram-positive or gram-negative and is more specific. An example is polymyxin B, which only kills gram-negative bacteria. And there are many examples, that is just one. Exercise 41, page 354, continued. Antibiotic resistance, what is it? Bacteria is unaffected by the antibiotic. In other words, you go to the doctor for your UTI, they prescribe you an antibiotic, and you don't get better. The antibiotic is unable to kill the bacteria. That is not a desirable outcome. This occurs when antibiotics are prescribed when they aren't needed. So in other words, if you have a viral infection, you do not need an antibiotic. In my previous example of a UTI, you would need an antibiotic because that is developed or based on a bacteria. So you only need to take antibiotics for certain infections, strep throat, UTIs, etc. This occurs when antibiotics aren't taken properly. So as the patient, if the medication says take twice a day for 7 days and you don't, you take it once a day for 4 days and then you discontinue the antibiotic, all you have done is killed the weak microbes. What is left in your body are the strong microbes, what we call superbugs. Those bacteria are then potentially passed on to someone else. So it is very important that you not only take the antibiotic for the amount it says, but how it says. It might say on the bottle, take with food or don't take with certain medications. That ensures that the antibiotic works properly in your body. But it's very important to not take an antibiotic unless you need it and to take it as prescribed. Healthcare settings have also attributed to antibiotic resistance. Overuse of things like triclosan, which we've talked about in exercise 40, over many, many years have led to antibiotic resistance. Superbugs. These are microbes that are super, but not super good. These are microbes that are resistant to many antibiotics. So you could take a variety of antibiotics and none of them are going to effectively destroy or kill the bacteria. And therefore, you could potentially die from a minor infection. Superinfections are infections that can be difficult or impossible to treat. It is possible now in this day and age, you could get a particular strain of Streptococcus pyogenes, which causes strep throat, and it can be life-threatening. It is possible. Page 355, 356. We're going to look at the setup of the experiment. This is called the disc diffusion method. This disc diffusion method is a fancy word to say we're going to be adding antibiotic discs to plates and seeing how effective the antibiotic is. This is similar to what we looked at on the antiseptic disinfectant. The only difference is we're using filter discs that already have in them the antibiotic. So you would inoculate 2 nutrient agar plates by thoroughly swabbing the surface with bacteria. Again, we are using E. coli and Bacillus for the same reasons listed in the previous presentation. E. coli is gram-negative and Bacillus is gram-positive, and spore former versus non-endospore former. You would let the plate sit for 5 minutes. You would add the antibiotic disc in the correct region of the plate. We're going to look at 8 different antibiotics, so 4 antibiotics per plate, and then you would incubate the plates for 48 hours at 38 degrees Celsius. You would see page 355 for details. Page 356. On page 356, complete and submit the purpose. On 356, complete and submit the hypothesis. See directions below. So what you're going to write is which antibiotic or antibiotics, you can pick more than one, that you think will be the most effective for each bacteria. Again, a hypothesis does not have to be accurate. So you could say, I think the most effective antibiotic for E. coli is fill in the blank, or for Bacillus. I want you to do one for each bacteria. You will also do this for the least effective. So you need a minimum of 4 hypothesis statements, one for each bacteria, most and least effective. The antibiotics are listed on the next page. You will submit this page, page 356, on Blackboard. Page 358. This is the same slide we saw previously when discussing the antiseptic and disinfectant effectiveness. The analysis is the same way. We would measure the zone of inhibition, and the larger the zone, the more effective, in this case, the antibiotic. These zones are also going to then be used to figure out whether or not an antibiotic is sensitive, intermediate, or resistant, which I will explain on the next few slides. Page 358, instructions. On page 358, fill in the method of action for each antibiotic. Most of those have been discussed in this PowerPoint presentation. Use this PowerPoint, the lab manual, and the textbook as resources. If I can recall correctly, there's only one antibiotic that we've not discussed that's on the list. Use the data on the next slide to calculate the averages for each antibiotic. And there's how to do your averages, which we've discussed previously. And then you're going to use those averages and use table 41.1 on page 359 to figure out R, I, or S, which again I will discuss with you on the next page or next slide. Okay, so here's page 358. So what we see first is a column of all kinds of antibiotics: ampicillin, bacitracin, vancomycin, penicillin, tetracycline, chloramphenicol, trimethoprim, sulfamethoxazole, and ciprofloxacin. So the AM10 or the B10 indicate the code that would be seen on the antibiotic discs and the amount of drug impregnated on the disc. So in other words, AM10, usually like 10 micrograms of ampicillin. You don't need to know that amount, just need to know the antibiotic. So then we have a list of hypothetical data, and again, you're going to average. So for example, you would add 23 plus 28 plus 25, divide by 3, and so on and so forth for all the antibiotics for E. coli and Bacillus. Once you have the averages for each antibiotic, then you're going to look at something called R, I, or S, and this is where you typically get a little confused. So let's simplify. The R stands for resistant, I stands for intermediate, and S stands for sensitive. So what you would do is use the table 41.1 and the lab manual and the averages to calculate or figure out R, I, or S. So when I have in that last column G plus and G minus, that means Bacillus versus E. coli, gram positive versus gram negative. So for example, let's say AM10 average, and I'm just going to guess, is let's say 26 or 27. You would take that number 26 average and look on table 41.1 and decide based on the table, is it intermediate, sensitive, or resistant? You do the same thing for E. coli. So what do you want to have? A lot of R's, I's, or S's? Well, you would want a lot of S's. S's means the antibiotic is effective, sensitive at killing the bacteria. I's mean they kind of work, and R's mean the antibiotic did not work. It was resistant to, the bacteria was resistant to the antibiotic, so we don't really want a lot of R's. The other part you're going to do with this table is answer some questions from analysis. One of the questions that I ask in analysis is to compare and contrast broad spectrum versus narrow spectrum. So how would you do that? Remember, broad spectrum are effective against gram positive and gram negative. Narrow spectrum are only going to kill one type of bacteria. So for example, if you saw numbers of zeros in one set of bacteria, and then there were positive numbers like higher numbers, 20s, 30s, etc., and the other bacteria, that would mean it would only kill that one type of bacteria. Last but not least, you should fill in the method of action. Most of the methods of action have already been listed in this PowerPoint presentation. The only one that we have not discussed that you'll need to research either using your textbook or the internet is bacitracin. Bacitracin is an antibiotic that's actually produced from the bacteria Bacillus. So this is table 41.1, and you can see that you're going to look at the resistant, intermediate, and sensitivity columns. The numbers that you see on this table, you're comparing to the averages for each antibiotic for each bacteria. So for example, if I were to have a zone of inhibition on penicillin that was 25, it would be S for sensitive because 25 is greater than 15. Another example is if I had on tetracycline for, let's say, E. coli, a zone average of 17, so tetracycline, a zone average of 17, it would be intermediate. Last but not least, if we had amoxicillin and my zone of inhibition average was 52, it would be R resistant because 52 is greater than 13. So you use the columns to try to find out resistant, intermediate, or sensitive. Exercise 41, page 358, 360. Complete and submit the averages for each type of bacteria on page 358. Complete and submit the methods of action on page 358. Complete and submit the R, I, or S on page 358, which was the previous table. You will submit all of this on Blackboard. Complete and submit the conclusion and analysis portion on page 360. So make sure that you have read through those questions and answered them thoroughly. Pages 361 through 364. Complete and submit the case studies on page 361 through 362. And you will submit this on Blackboard. These pages actually go through hypothetical scenarios. So patients with different illnesses, you're going to use the table for RRIS, sensitive, intermediate, and resistance, and the antibiotics discussed in this presentation. You will also need to watch the history of antibiotics video. This video is about 1 hour that we would normally watch in lab. You're going to answer the questions on page 363 through 364 as the video is playing, and you will be submitting this page. These questions will also be on the lab quiz. The history of antibiotic video link is found on Blackboard. You may need to enter your data or your user and password through ICC. If you can't get the video to work, contact your lab instructor. This video is from the History channel. It's called Modern Marvels, and it goes through a very interesting display of, or interesting description of antibiotics. Last but not least, it is important to take the lab quiz on the mastering site via Blackboard. This is the end of this exercise.